Good afternoon, sir.
Just start calling.
Good afternoon, sir. Good afternoon, madam.
Good afternoon. You guys had lunch?
Yes, ma'am.
Yes, madam.
Okay.
Yes, ma'am.
Actually, it was informed that today there is a quiz.
Yeah, there will be a quiz. Let me check. Yes, there is a quiz at 5 to 6 p.m. Okay. Quiz session
Okay.
is
Will
5
it be
to 6 p.m. Yes. This session will be over at 4 o'clock. Then we'll have a break. And then 4 to 5 there is a break. And then 5 to 6 there will be a quiz. Okay.
in
Quiz.
Google Forms, ma'am?
In Google Form, that I really don't know. ये quiz जो है वो Google Form में है क्या?
Yes, ma'am. It was informed that it was in Google form only, ma'am.
Okay, maybe yes, that will be Principal Mehboob Nagar, Mr. Bhaskar sir will be conducting it. Okay, he will join us at that time and we will get to know. There is online written quiz. Google Form में ही होगा.
Yes, ma'am. Bhaskar Kumar, sir.
Yes, yes, yes.
Madam Bhup Singh joined?
Yes, sir. Yes, sir. Bhup Singh sir and Nirusha ma'am both are here, sir.
ability to code the chemical problem to नहीं बना.
सही चार्जिंग बैठा चार्जिंग बैठी
कल भी इधर ही करना है तुमको फॉर्म.
हाँ
इधर करना है.
नहीं वो 5 है क्या मैम 16 हुआ
हम दे दे और थोड़ा पावर लाते हुए इसे लगाना
एक बार हमने तुम देखा तुम्हें सुन रहे क्या.
नहीं मैम कुछ नहीं सुन रहा
बोल रहे हैं.
वो कुछ नहीं बोल रहा है इसलिए सुन रहे कोई माइक ऑन नहीं
माइक ऑन नहीं है.
एक बार चेक कर.
नहीं वो कोई और को बोल रहा है किसी और को बोल रहा है
सुन गया ना.
नहीं हम हमारे
सुन रहे हैं ना इसमें आवाज आ रही है.
नहीं
उनकी आवाज
नहीं नहीं
म्यूट.
हाँ वो म्यूट है
Madam, sir has guest has joined.
Okay, sir. Okay, sir.
कुछ issue आए practical को मिले।
Welcome, Ashwin, sir. So, students, today now in our session 3, we are having Dr. Ashwin Dalal. Sir is the head of Diagnostic Division, Center for DNA, Fingerprinting and Diagnostic, Hyderabad. And sir is he's in MD in Pediatrics from Goa Medical College, Goa University, DM in Medical Genetics from Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow, India. Dr. Ashwin Dalal is a recipient of gold medal in his MBBS exams. Sir did his post-graduation in pediatrics from Goa, as I told you. After working for 1 year as a senior resident in Goa Medical College, Sir joined the DM course for super specialization in medical genetics. And during his DM course, he gained expertise in the fields of clinical genetics, molecular and biochemical genetics, and fetal medicine under the able guidance of Dr. Shubha Phadke. He has published a number of papers in national and international journals. After completing DM in medical genetics, he joined Center for DNA, Fingerprinting and Diagnostics in April 2007. His interests include dysmorphology, neurogenetics, and fetal medicine. We all welcome you, sir. Ashwin, sir. Thank you so much for being here, sir.
Thank you, madam.
And above all the introduction, I am very happy to say that Dr. Ashwin Dalal is a Navodaya student, ex-Navodaya student from JNV Goa, South Goa, I think.
Yes.
Am I right, sir?
Yes.
Yes. Thank you. Welcome, sir.
Yeah.
Most welcome, sir.
So very good afternoon to all of you, and I hope the Science Congress is progressing well, and the principal from JNV here requested me to talk to you, so I'm very thankful to Mr. Daniel for inviting me. It's always nice to speak to all the students. I also was in their place at some point of time, and so today we'll have a discussion about the topic that you must have seen, because I went into genetics, so I'll try and share with you in very simple terms how we use the principles. These are all simple principles which you must be learning in your biology textbooks, and I'll just try and show you how we use them for patients with genetic diseases. We'll discuss a little about the ongoing pandemic, how genetics helps us in testing for this viral presence, and I'll try and share with you some memories of JNV 25 years back almost that I spent in the school there. So we'll begin with our Are you able to see the screen?
Yes, sir.
Okay. And the audio also is okay, no? Yeah. Okay.
Yes, sir. Yes, sir. Yes.
Yeah. Okay. Okay. So today we'll discuss about the story of ATGC. So you know ATGC are the nucleotides. It is the genetic code of life. The whole life depends on right from the small virus or bacteria up to the highly evolved organisms like humans. All life is coded by these nucleotides, that is adenine, thymine, guanine, and cytosine. So I've just tried to put that in form of a story for all of you to understand how these nucleotides really shape our life and what they decide or how they lead to some problems which we need to solve then. So, so before we start, I would like to congratulate all of you on the 75th independence year of our country. So we are having a lot of programs under this Azadi Ka Amrit Mahotsav initiative of Government of India, and we all should be very proud to be the citizens of this country where a large number of people have really strived to achieve this independence, and we are enjoying this 75 years of independence, that we are able to study what we want to study, work where we want to work, do whatever we please instead of having any problems from anybody. So we have to value this and always remember the hard work put in by all these great people, and try to do your best for whatever you can do for the country. Okay. So now I have broken this talk into 4 stories which represent the A, G, T, and C. So let's see the first story, that is about a letter A, and it tells us about a book of life. Okay, so if you look at our English language, then whatever all the literature or all the books in the English language which are written, millions and millions of books are there, all of them just use these 26 letters, and using combination of these 26 letters we make all the words and all the books that are published throughout the world. Similarly, our human genome, instead of using 26 letters, it just uses these 4 letters A, T, G, C, and different combinations of this A, T, G, C is present in each and every cell, and each of our cell contains 3 billion of this ATGC. So you can imagine how many nucleotides are present. So this 3 billion bases in each cell decide the fate of that cell, what protein it is going to produce, what function it is going to do. It is all decided by the 3 billion bases, and all these 3 billion are made up of only just these 4 letters A, T, G, and C. So just like we can use this 26 letters to write different kind of books, similarly our genome with this ATGC combinations creates all different types of people throughout the world. So we have different types of people, we have differences among ourselves, you are different from all your classmates, and that is just because of the differences in the way the ATGC is arranged in your cells. And just like in English language also, you make sometimes a spelling mistakes, but these spelling mistakes lead to a lot of problems in interpreting. Similarly, our genome also sometimes makes mistakes, but the problem is that this mistake can be solved by just rubbing or replacing this particular board. But this one mistake in our genome, so just one mistake in 3 billion bases, so such ATGC there are 3 billion, but just one change from A to T is not tolerated, and just this same change can lead to a disease called sickle cell anemia. You must have heard of this disease, where this change leads to amino acid change from glutamine to valine, and that leads to a lot of problem with our red blood cells, ultimately leading to large number of problems with the patient. So you can imagine what is the importance of our genetic code and how accurate it needs to be, that it is not able to tolerate even one spelling mistake at a particular nucleotide. So how do we go about finding these mistakes? So whenever there is a patient who is suffering from a genetic disease, we go about finding these mistakes by sequencing the genes. So in the laboratory, we take the DNA and we sequence. So this is how the sequencing result comes out. So we have A, T, G, or C. So in every part of the genome, this sequence can be checked. So if we want to check at a particular place, we can see here that this particular patient has, this is the normal, but instead of this G at this position in the patient, it is a T. All other nucleotides are same, just like the normal person, only instead of G, this person is having T, and hence the disease is occurring. So in this way, we can check for the mistakes. So this is same like if you know that there is a mistake in one edition of the book, then if you want to find the mistake in the second edition of the book, you don't have to search the whole book. You can know which page number it is, go to that page number, and you will be able to find the mistake. So similarly, if we know what we are looking for, then we can just sequence a small part of that gene and check whether it is a G or T at that position. Okay, this we can do if we know where is the mistake. But if we do not know where is the mistake, so if we do not know, a patient comes to us and we just need to find out what is the problem with that patient, then finding that one mistake is something like looking for this dot in all volumes of Encyclopedia Britannica. So that is what I was telling you, that our genome is 3 billion base pairs. So 3 billion means 1 billion is 100 million. So 3 billion bases we have to check for this one mistake. So that is very, very difficult to do. It is like finding needle in this big haystack. So you must have heard of this term, finding needle in a haystack. So it is equivalent of that. Just this one needle has fallen somewhere here and you have to find it. So it is a really difficult task to find this one single mistake in a huge amount of DNA that is present. So what do we do? So if you have to find needle in a haystack, this is not really the way to find. If we go this way, then we are not going to find our needle. So what do? We take the hay, then this haystack is divided into multiple small blocks. We take one block at a time and we'll find so many needles, but we want to find our specific needle. So we'll use some sort of a sieve to find this needle. So this is an example that you will find the needle in the haystack. Similarly, we use a bioinformatic pipeline. So this is a computer based. So the whole field of genomics or genetics now is a lot computer based. So we need to have information both about the biology as well as the computers, because humanly it is not possible to do this kind of calculations. So we have a complicated pipeline through which the data is generated and it is analyzed to find that particular one mistake which is present in the particular patient. So now through sequencing of this DNA, it is possible to sequence the whole genome of humans. So all 3 billion base pairs, now it is possible for us to sequence. And what is the cost? So the first human genome costed almost $2 billion. Okay, in rupees, it will be crores and crores. But now it has come to the position that now if we want to sequence a whole genome, we can do it for as less as 60,000 rupees. So this drastic decrease in sequencing has happened with newer and newer technologies. And this genome can now give us a lot of information about the genetic diseases as well as about the probability of occurrence of diseases in future. And this is a huge area of study where people are trying to find out from the genome whether you will develop diabetes or hypertension or coronary artery disease when you are an adult. So we are able to, because as I told you, this genome decides what proteins are formed in the body, and those proteins are going to lead to all the functions, and any abnormal function can come leading to these diseases like diabetes, cancer, hypertension. So people are trying to predict these diseases based on the genome sequencing. So when you have so much power that you can predict future, you can predict whatever is going to happen to a patient, then you have a lot of responsibility also. So every good thing can be used in different ways. You have the great power, but with that you have great responsibility also. So this is your friend Spider-Man also has told that. So what is our responsibility? Our responsibility is that we should not be using this kind of power to create designer babies. So you must have heard a lot of people wanting to try to create designer babies like with high IQ, without any disease, with good athletic skills or perfect vision. So these kind of things should not be done. So every great invention, every great discovery has its positive and negative side. You must have heard about electricity. You can use it for a lot of good things, but it can also give you an electric shock. So you need to decide how responsibly you can use this great power for betterment of humans. Okay, so that was about the letter A, which told us about the book of life, how our life is coded by the ATGCs. Now let's go on to another letter, that is the letter G. So what is this letter G telling us? This, we take an example of a goldsmith. So if a goldsmith is sitting and he wants to buy some ornament from you, then if you see all ornaments look the same, so how is he going to decide which is real and which is fake? Okay, so what does a goldsmith do? So he will either use his visual observation skill, or he may use some machine like a carat meter which can tell him how pure it is, or finally he will melt the gold and then he can make out what is the purity. So this is what the goldsmith will have a strategy to decide whether it is a real or fake. A similar problem is faced by all geneticists, or in genetics we face this problem because, as I told you, we have 3 billion bases in our cells. However, there are differences among these bases among different individuals, and that is what makes us different. So these differences are called as polymorphisms because they do not cause any disease. It is these differences which make us different from each other. You are different from your brother, you are different from your friend, colleague. So these differences at multiple places in the genome make us different from each other, but they don't cause any disease. That's why we call them as polymorphisms. Whereas those differences which lead to disease, they are called as a mutation. So it is very difficult for us whenever we find a particular difference, we have to differentiate whether it is a disease causing pathogenic variant, that is a mutation, or it is a benign non-pathogenic variant or a polymorphism. So just like the goldsmith, we have to solve this dilemma whether it is a disease causing change or not. So like goldsmith uses different strategies, we also use different strategies to decide among the two. So what do we do? So let's see. So we have this one particular patient who has different clinical features and his x-rays are showing some conditions. So based on the clinical features and x-rays, we diagnose that this patient has this one particular condition, mucopolysaccharidosis. Okay. Now this is caused due to deficiency of a particular enzyme in the body. So we do the enzyme level and we find that the enzyme is low in the patient. So this is one level of evidence that this patient has this disease. Next, as I told you, this enzyme deficiency must be occurring because there must be some change in his DNA. So we go ahead and sequence this particular gene which that enzyme is coded for. So we look that in normal it is a G, whereas in this patient it is a A. All other sequences are same in normal and patient, only at this position instead of G he has a A. So now we have the mutation which is responsible for this, but now we have to know whether this change is leading to this disease or not. So that is where the Goldsmith strategy comes, that we have to decide whether this is leading to that disease or it is a normal polymorphism. So we use different software where it tells us whether it is damaging or not damaging on a scale. So different softwares are used. Then we use some protein modeling. So just like we have the biology proteins, we can do the same in computer. So we can make the model of the protein and change that particular amino acid and then see what happens to the neighboring amino acids. So we can do protein modeling. However, if we want further evidence, like always we want more and more evidence. So that is true in science, that is true in medicine, that we want to be absolutely sure. So we want more evidence. So for more evidence, then we go to the model organisms. So we use different model organisms like zebrafish. This is the fruit fly which is called as Drosophila. We use the mouse model. We use the worm, that is C. elegans, or also the primate monkeys are used as models for studying human diseases. Also, we can make pluripotent stem cells. So you must have heard a lot about stem cells that are being used for treatment or for diagnosis. So these stem cells are cells which are taken from the patient. So we have different kind of cells in our body. So we have skin cells, we have brain cells, we have liver cells. So these all cells have different functions. So now what we can do is that we can take the skin cells from the patient and convert them into either neurons, or if you convert them into heart cells, then you can see the cells start beating. So that is the work of the heart. So these cells are taken from the patient's skin, then using certain transcription factors or chemicals, we convert them into heart cells. And once they are converted to heart cells, they start beating. So once they start beating, we know that they are now converted into a heart cell. And now we can use any particular new drugs we want to test, then we can use on this cell line and study that. So in this way, different kind of modelings are used to study human diseases or new drugs that are developed. So that is about how we go about finding difference between a polymorphism and a mutation. Now let us go on to the third letter, that is the C. So this is telling us about the care to cure. So can we treat these genetic diseases? You must have heard that many of these genetic diseases are not treatable. It is very difficult to treat because this change is present in each and every cell of our body. So it is difficult to go into each and every cell and do treatment. So what is the normal medical paradigm is that you go to a doctor as a sick person and doctor gives us some medicine and we become all right. But in genetic diseases, that is not so. A lot of these diseases are not curable or many of them do not have treatment. So we try to do some sort of a genetic counseling where we inform the patients about what treatment options are available, if any. Then we tell them same disease can happen in another child because all these diseases follow the Mendelian pattern of inheritance. You must have heard this in biology, autosomal recessive, autosomal dominant. So these kind of inheritance patterns are seen in these diseases. So the second child also can be at risk of developing this disease. So we do prenatal diagnosis. That means we check the baby while the baby is in mother's womb, whether the baby has that particular mutation or not. And if baby has the mutation, then they go for termination of pregnancy. If baby does not have, then we continue the pregnancy. In this way, the family can be happy by having a normal child. Newer and newer genetic therapies are also coming up with lot of research. We have different kinds of therapies that is coming up for different kind of genetic diseases. So we really can't say now that they are not at all treatable. So at different levels, people are developing therapy. Like at the gene level, when you do therapy, it is called a gene therapy. At the RNA level, we can do at the protein level by replacing the protein. At the metabolic function level by doing dietary or pharmacological intervention or clinical phenotype. So we can do some surgery for the heart disease or something. So in this way, different levels of therapies can be done for the patients. So now since therapies, we have discussed about the mutations, the disease causing variants and the therapies. Now let us see the letter T. This is the last letter which is telling us about how the traditional medicine is meeting the modern medicine. So our traditional medicine, that is the Ayurvedic medicine, we have this concept of Ahar, Vihar and Aushadhi. That means our ancient gurus, they did not only prescribe medicine, they also prescribed the lifestyle and the diet. Okay, so it was a combination of all that decided what treatment is to be given for the patient. So each patient, the Ayurvedic doctor will check in different ways that they had, and then medicine will be made for that particular patient. On the other hand, our modern medicine just gives one treatment for all diseases. That means if you have fever, everybody will be given paracetamol. If you have another particular pain, everybody will be given the same medicine. So this concept of personalized medicine is something which is coming up, and you will be reading this in different paper articles or somewhere, where the present system is like one size fits all. That means same medicine can be given to all, but the personalized medicine says, like our age old traditional medicine, the treatment should be right treatment to the right person at the right time. And this is very important, and this is going to be the future of medicine. So let us take this one example to explain to you why this is important. So if you take, these are the 100 patients with a particular disease, that is a blood cancer in children, and we use this one particular drug called as 6-mercaptopurine. Okay, This is a medicine which is used for treatment of this patients with blood cancer. Now what happens, we have in our body an enzyme called as thiopurine methyltransferase. So what this enzyme breaks down this drug, and then this drug is excreted in urine, so that the drug does the work and then is excreted in urine. Now what is seen in normal people is that about 88% of the people have good enzyme activity, and hence they are able to metabolize this particular drug, and the drug is excreted in urine. About 11% of people that are shown in yellow here, they have low activity of this enzyme. So because of low activity of this enzyme, this drug remains in their body for a longer time, and hence these people get lot of side effects because of the drug. Okay, and this 0.3% people, so this is 1 out of this 100, he doesn't have any activity of this enzyme. So because he has no activity, this drug will always remain, it will not be removed from his body, and he will die because of the drug. So instead of dying because of cancer, he will die just because you gave the drug to him. Now if we have to avoid this, what we can do is that instead of giving same dose to all of them, we can give normal dose to these people, and we can detect these people who have the less activity of enzyme, these yellow people. If we detect, we can give them a low dose of the drug, and this one person we should give some other drug and don't give this drug. So if we are able to do this, this is what is personalized medicine. That means right treatment to the right person at the right time, and that way at least we will not have a lot of side effects, or that one person will not die because of the disease. And this is just one example, but we see this for a lot of drugs like hypertension drugs, heart failure drugs, antidepressants. All these drugs, this is seen that all people do not respond to the drug in the same way. Some people respond better, others don't respond better. So for all these kind of drugs, in future there is going to be a test which will be done, and then only the drug will be given. And already for few drugs now, it is already compulsory that we have to check for the genetics of that individual, and then only the medicine is given. So this is going to be the future of medicine, where hopefully in the future, the predictive medicine at the birth of baby, we will be sequencing all the genes of the baby. We will get to know all the proteins and the metabolites, and then save that in form of a credit card kind of a small chip. And then whenever you go to the doctor can insert this chip in his software and get to know what are all the drugs that you are allergic to, what are all the drugs that should not be given to you, or which disease is likely to happen in you in future. All this information is going to be slowly captured and used by the doctors. So in future, we will shift from using the traditional patrika for matchmaking, and we'll probably using a gene patrika or a genetic horoscope to decide what can be the best match so that you have a healthy babies. So these were, in brief, the four stories that I put together. I hope I have been able to tell you in brief about the importance of genetics, how genetics is used in patient treatment, patient management, how we make use of the same principles that you learn in your textbooks to patients directly. Okay, so now we will shift to another topic, that is about the ongoing pandemic. So that is the COVID-19. So we all know that this COVID-19 is caused because of this one particular virus called as SARS-CoV-2. That is the name of the virus. And this is the structure of the virus. You must have heard a lot about this. So this virus has a membrane glycoproteins, nucleocapsid proteins, the RNA is present inside the virus, the envelope proteins, and the spike proteins. So these are the spike proteins, and you must have heard that many of the vaccines are made against these spike proteins because these are the proteins which interact with the human cells. So what happens when this virus enters human body? So these spike proteins are present on virus, and this for these spike proteins there are receptors in our cells in the throat. So these are ACE2 receptors. So when this virus enters in our body, the virus comes and attaches to this ACE2 receptor, and then it enters our body. Once it enters our body, it leads to all the different symptoms of fever, dry cough, breathing difficulty, tiredness that leads to this COVID-19 disease. So there are different ways in which you can diagnose this. You must have heard of all these. You can do in multiple ways the antigen test, the antibody test, or you can do the real-time PCR. Also, you can sequence the genome to know which type, like is it a delta, is it a alpha, beta, or the new Omicron, which kind of virus it is. We can identify doing sequencing of the virus. So the most commonly used technique is the real-time PCR, and that is what we also use in our laboratory to detect the virus. So how diagnostic testing is done for the virus? So you must have heard about this sample collection. So we collect sample using a swab here, which is put in the nose because we need the nasopharyngeal sample. So the swab is put in the nose. Then the swab is put in a viral transport medium, okay, that keeps the virus alive, and it is transported to the laboratory. So in the laboratory, we take this medium which is mixed with the virus and we isolate the RNA. So we take out the RNA from this medium, then this RNA is converted to cDNA, and this cDNA is used for doing a real-time PCR. So real-time PCR, we get these curves, and based on that, the reporting is done to the clinician whether it is a positive or negative for the particular virus. Okay, so this is the setup of the laboratory that we have in our institute. You can see it's a large amount of meticulousness is required because this virus is very infective, and we have to make sure that the people who do the testing should not get infected. We have to follow all aseptic precautions. So we have to have a separate area where the live virus is handled, and then it comes out, and then it is a killed virus for doing this other steps. So how it is done actually? So you can see these all people who work with the virus, they wear something called as a PPE, that is a personal protective equipment. So it includes a full body gown here. Then they have shoe covers here. They have gloves. They have face mask, N95 mask, and they have goggles. So when you put all this is called as the PPE or the personal protective equipment. And also we use the laminar flow hoods which prevent the air from coming out of this, so the person is protected while working with the virus. So this is the initial step where the viral sample is taken out, and then we do RNA isolation using different methods. Centrifuging, we take out the RNA. So once the RNA is taken, now the virus is inactivated. So it goes into the next laboratory, that is the real-time PCR. So a real-time PCR is done and then analyzed on the computer to detect whether it is present or not. So how the real-time PCR results look like? So you can see this is a negative sample. So here we have the curve only for the internal control. Now internal control means a human RNA. We want to detect is there any sample in that swab or not. Sometimes they may not put the swab inside the nose properly. Then the internal control will not be there because there is no human RNA. But if internal control is there and the viral curve is not there, then we can say this is a negative sample. Here you can see the internal control is there, plus we have these two peaks for these two genes of the virus. So E gene and RDRP gene, these are the genes of the virus. So because these curves are present, we will say that this is a positive sample for COVID-19. And looking at these curves, we can also detect the number of viral particles. So if the curve starts at a higher number of cycle, like these curves are starting at beyond 30, that means the virus number is less. So this is sometimes called as the CT value. You must have seen these reports where laboratories give the CT value. So the CT value here is 30. That means after 30 the virus is detected, whereas here the CT value is almost 22 or 23. So this patient has much more virus load compared to this patient. So in this way we can calculate the viral load in the real-time PCR. Okay, so just we will go through this brief video about how the exact testing happens in our laboratory.
The Center for DNA Fingerprinting and Diagnostics, CDFD, is a premier research institute under the Department of Biotechnology. CDFD is located in Hyderabad in the state of Telangana. It conducts research in various disciplines of life sciences and also provides services in fields such as forensics, diagnostics, as well as basmati rice analysis. COVID-19 has been recognized as a pandemic in March 2020, and CDFD has been one of many testing centers approved by ICMR and DBP for COVID-19 testing. Sanitization of hands is indeed the need of the hour. With a team of specialists trained in molecular biology, CDFD has been able to conduct testing of COVID-19 from patient samples brought from hospitals across the state of Telangana. This is the COVID-19 testing laboratory. Each worker entering the lab must follow biosafety rules by wearing a lab coat, gloves, N95 mask, as well as shoe covers. PPE or personal protective equipment is donned before entering the biosafety lab or BSL. These one-time suits ensure complete safety to the worker from head to toe. The first step in the testing process involves patient sample handling. The nasopharyngeal swabs, which are brought in vials from hospitals, must be handled with extreme care. These vials are brought in boxes, which are first sterilized and then unpacked. Individual packets containing these vials are then taken to the biosafety cabinet. Here, the patient sample is inactivated using specific reagents. The biosafety cabinet has been designed in a manner to create a protective workspace to ensure complete safety while handling these patient samples. All biosafety measures are followed strictly in this area. A portion of the patient sample is also stored in deep refrigeration for future research or in the case of repeat testing if required. The second step of COVID-19 testing involves the RNA isolation. Inactivated patient samples are brought from the BSL facility to the RNA prep room. The RNA prep room also comprises of two biosafety cabinets to ensure safety to the workers. Here, the inactivated patient sample is taken for RNA isolation using specific buffers and reagents. This is done either manually or by using an automated machine for quicker processing. The RNA prep is now eluted for downstream processing. The third step is the identification of specific COVID-19 genes from the isolated RNA sample. This is accomplished by a technique called as RT-qPCR or reverse transcriptase quantitative polymerase chain reaction. The presence of COVID-19 emits a fluorescence which is detected by the software and then analyzed. The final step is the documentation, where the results of positives and negative cases are documented and then sent to the government agency on their online portal. CDFD also plans to engage in COVID-19 research by sequencing the viral genome using the next generation sequencing platform. This is a part of their National Genomics Center facility. CDFD's dedicated volunteers are contributing to the national effort to combat the COVID-19 pandemic.
So I hope you are able to see the video and try and understand how the whole process of testing happens. So still till now also we are continuing doing this testing as well as sequencing for the different new variants that keep coming. So this was in brief again about the genetic testing for the COVID virus. In the next few minutes, I will try and talk to you about my journey from Jawahar Navodaya Vidyalaya to the present time here. You must have heard that Jawahar Navodaya Vidyalayas in the history. You must have heard that they started
Sir?
in. Hello. Yes.
Sir, 1 minute, sir.
Yes.
Sir, you said no, sir, that CDFD is wearing PPE kit and testing the laboratory.
Yes.
But at PHC centers, it is not following, no, sir.
What is it?
Rural areas, the
Hello?
people who are testing the patients not at all wearing the PPE kit, no, sir.
Not the
Yes, sir.
patients. The technicians should wear the PPE kit. The technicians who do this.
Yes, sir. I am also saying that only, sir. Technicians.
If they are not wearing, they are doing it at their risk. They should wear the PPE. So
yes, sir.
Yeah, so it has to be worn by whoever does the. Otherwise, they are at risk of developing this disease. No? So they should be wearing the PPE while doing the testing. So we follow it in our laboratory. Yeah. So I was saying about if you heard about the history of the Jawahar Navodaya Vidyalayas, the first two schools were started in 1986. One was in Haryana and the other was in Amravati. And in the year 87, almost 80 more schools were started throughout the country. And that was the year when I entered into Navodaya Vidyalaya. So ours was the first batch in 1987. So I come from a small state of Goa, and this is the place called JNV Kanakona where it is located, the Jawahar Navodaya Vidyalaya. So this was in 1987. So I have a lot of nice memories of the school days. So we had both our extracurricular activities as well as the curricular activities, a lot of encouragement. That time it was really the beginning of the Navodaya Vidyalayas, the first year. So everybody, even the teachers, the students, everyone was new, and we were trying to adjust to this new kind of teaching system that had come up that time. So you can see this is the 1988 photographs that I could take out to just show you that where all it started. So I completed in 93. So this is just to show you, because it is a science exhibition. So I remembered that we also used to have these science exhibitions, and these were the two models that I had made during the science exhibitions during my 8th and 9th standard. We used to go to different Navodaya Vidyalayas and take the models with us and then explain. So that time it was this SLV-3 from ISRO, and then the INSAT-2B. This is the satellite which was launched at that time. So this was just a nice thing to remember those days. Today, now also you must have made some models or have the elocution competitions on science and different aspects. So it's a nice way to get into this kind of small things which in future are really going to help you to increase your scientific acumen or your interest into doing things. So after my 1993, I went on to do MBBS, and I was fortunate to have the gold medal during that MBBS. Then I did MD in pediatrics. So this is child specialization from the Goa Medical College Hospital. So after MD pediatrics, like any other medical student, I thought I should try private practice. So I started doing some private practice, but could not find any interest much in that. So then I went further to SGPGI MS Lucknow. This is Sanjay Gandhi Postgraduate Institute in Lucknow. And there I did a DM in medical genetics. So at that time, this was the only place in the country where DM genetics was available, and only 2 students they used to take per year. So through an entrance exam, I got in there, and that is where I got exposed to the research aspect of medicine. So a lot of this genetic research that goes on. So all that I was exposed to, and with all that thing, then I shifted from a medical background to a scientific background. So I joined as a scientist in this institute called as the Center for DNA Fingerprinting and Diagnostics. So here we do patient evaluation for various genetic diseases. We check them for what genetic diseases they have. We do genetic testing for these diseases, as well as we do research mainly in the rare genetic disorders. We try to identify what are the genes which are causing problem to the particular patients. And our work over almost last 15 to 16 years, it has resulted in a large number of publications. We have identified about 5 novel genes for various genetic diseases, and this was covered a lot in various newspaper items and all. This is Dr. Venkaya Naidu, who visited us, our vice president visited recently our institute. So I'm telling you all this mainly to encourage you that I was also a student just like you in one of the Navodayas, and with your willpower, with your efforts that you will put into your studies, your extracurricular activities, your exposure to different exhibitions or whatever you do, always try to do the best in your life, and you also should be able to achieve great things one day. So I hope all of you become good citizens of the country, do something for the country. What our country has is helping us this 6 years from 6th standard to 12th standard. It is investing in us to become good citizens of the country, and I hope all of you are able to do that in future. So thank you very much. I'm really happy to be talking to you on this occasion, and to my, I should say, schoolmates, because all JNV, I even recently had a PhD student who had come from JNV. So JNV has expanded so much. Like when it started, it was just few schools throughout the country, but now in so many different aspects of life, like I have friends who are IAS officers, I have friends in different police or all sorts, and there everywhere, wherever you go, you have to do the best in whatever field you go. That is the whole hope of having these special schools, to train you into educational as well as into co-curricular, to make the good citizens of the future. And I hope that all of you will do great things in your future. So all the best for a great successful life to all of you. Thank you.
Thank you, sir. Now students can ask any queries. If you wanted to ask Dr. Ashwin any career guidance questions or anything related to STEM activities, you are free to ask him.
Sure, I'll be happy to answer any queries that you have. Don't feel shy. I told you I was just like one of you only. So you can. No question is good or bad. No question is simple or not so interesting. You can.
You can ask anything if you want me to. Nambur, if you have any questions? Yeah, JNV Nellore, yes.
If possible, you can put your video on. Yeah, yes.
You are not audible, Vita.
Unmute yourself. You can type the questions in the chat box also if you would like to do that.
Sir, I have a question, sir.
Yes, yes, please.
Sir, these genetical disorders are caused, are moved by parent to the children, sir. To stop this forwarding of movement of these disorders, what steps can be taken by people, sir?
Yeah, so that is very important to understand that genetics comes from the parents only. So we have, all of us, we have two copies of the genes, and one of those 2 copies comes to the child, one from mother, one from the father. So how we can prevent this genetic diseases from happening? So we can we try to do it in two different ways. One is called as primary prevention, one is called as secondary prevention. So secondary prevention means the first child is affected with some genetic disease, so we try to find what is that particular mutation in that child, and during the second child we try to check in the mother's womb itself whether this baby is affected or not, and baby is affected they go and terminate the baby. If baby is normal then they continue. So that way we can ensure that a normal baby is born to the couple. So that is called as secondary prevention. Primary prevention means first baby itself we want to prevent birth of a child with genetic disease, and that we can do by doing screening for various common genetic disorders. So we can screen for these disorders in population. We can check whether any of these couples are at risk of having a child with genetic disease. Various different tests are available now, and in that way we can then find out whether this couple is at risk, do the prenatal diagnosis, and that way prevent. Similarly, we have to also know that many of these diseases they happen because of marriages in relationships. You must have heard about this. So when there are in relation
Yes.
marriages, then there is a little higher chance. So if there is any such a marriage happening, maybe we should try and advise them that it's better to avoid such marriages, because the gene pool remains the same, and then autosomal recessive diseases are more likely to happen. And now we can even check for them. So it's best that the people approach the geneticist before planning for a pregnancy, and then we are able to prevent these occurrences.
Yes, sir. Thank you.
Yeah. Okay.
Sir, can I have a doubt?
Yes, please tell your name and which JNV you are from and ask the question.
I am Subhash Reddy from JNV Guntur.
Yes.
Sir, what is mercaptopurine?
So 6-mercaptopurine, as I told you, it is a drug. It is a medicine which is used for treatment of a disease called as acute lymphoblastic leukemia. That is a blood cancer. So blood cancer which is present in the children mostly. So this is the medicine which is used for treatment. So it's a drug which is used for the treatment. So that is what I told you about that particular drug, how it leads to different phenomena, that some people are not able to metabolize this drug and excrete it in the urine. So it remains in their body. That's what we try to check then.
Sir, and also what is genetics dilemma?
What is that?
Genetic dilemma.
No, dilemma means you don't know what is true and what is false. So I told that in the in relation to this differentiation between a disease causing mutation and a normal polymorphism. No, so we, as I told you, we are all different from each other. You are different from your friend. You look different. You have different habits. All these comes because the genes are different at some positions, what we call as polymorphisms. These changes do not cause any disease. So when we find a change in a patient, we have to differentiate whether this is a normal polymorphism or a disease causing mutation. So that is the dilemma what a geneticist faces, and then we use different methods to solve that dilemma.
Sir, what is paradigm?
What is it? Paradigm is just an English word. No, paradigm means a way in which you think of a particular thing. So like what's the paradigm now for a particular, what is the best practices followed, or what is particularly done at this time. Then if after 10 years if the things change, we will call it as a paradigm shift has happened. Then the paradigm is different at that time. So it's just an English word. It's nothing specific about genetics in there. Yeah?
Yes, sir.
Yes. Yes.
Sir.
Can you speak loudly? Who is this? Vishnu. What's your name?
Kishore.
This girl was. Please unmute yourself. Who is this?
Okay.
JNV Krishna. Who is it?
Kishore from JNV Pucheri, sir.
Yes, sir. JNV Krishna. Trisiras.
Yeah. Please go ahead.
Sir, is there any way to stop genetic disease permanently, sir?
Yeah, that's what, as I told you, no? So if we are able to do this kind of testing for everyone, the problem is that most of these testings are available only in big cities. They still have to go to different villages and all, because all the live births or the babies are born in villages and small towns. So if in future we are able to bring this technology to the smallest of the villages everywhere once it becomes available, then surely we should be able to stop these genetic diseases at some time in future. That is the overall aim. So we are working towards it. We try and see if it can happen sometime in future.
Sir,
Thank you.
Yeah.
sir, sir,
Yeah, please identify yourself so that we can check here.
Kishore from Jain Vitucheri.
Yes. Yes.
Sir,
Please.
genetic disease. Sir, how to prevent the genetic disease?
Sir, please unmute yourself, sir.
Yeah. So just now we discussed, no, about so we discussed just now for 2 questions, no, about how to prevent genetic diseases. Yeah, go on, please.
Excuse
Sir,
me.
I'm Devika from JNV Malappuram.
Yes.
According to you, what all steps can be taken to improve the medical field in India?
What is it?
What all steps can be taken to improve the medical field in India, according to you, sir?
Oh, so medical field in India basically is going to improve if you have more and more doctors who go to more and more villages. What happens nowadays, and maybe some same thing may happen with your friends also, that most of the people want to do this very high level of education, and then they don't want to go to villages, no? They remain in cities. So all the good medical facilities, everything remains to the cities. So unless it reaches the villages, there is no going to be a health for all. So the aim of health for all can be achieved when more and more medical facilities become available even to the smaller and smaller villages. So hopefully in the near future, as more and more good facilities come to the villages, there will be good facilities, medical facilities and doctors available in the villages. That will lead to the overall improvement in medical field.
Sir, I am Madhu from JNV Prakasham 1, sir.
Yes.
Sir, can we manipulate the genetic blueprint of an organism, sir?
Yeah, so that is possible now. So that is what is called as gene therapy. So you can replace one nucleotide with another nucleotide. You can change the amino acid. You can change the particular gene. And so in gene therapy, that is what is done. One particular mutation that is present in the patient, you replace it with the normal nucleotide, and then the patient can get better. So this manipulation is possible. That's what I told you. Whether this manipulation is used for patients to make them better, that's a good thing. But if you start using this manipulation to make designer babies, to make you more athletic and all that, that becomes a ethical dilemma. So that is where the problem is. But otherwise we are able to do that, and in future we are going to be able to do it for many more diseases.
Sir, sir.
Yeah.
So this is Nihira from JNP East Godavari, sir.
Yes.
Sir, where do the test developers get the RNA needed to validate the test performance for FDA?
For what?
For FDA, sir, where do the test developers get the RNAs?
So RNA, usually we will store it at minus 80 degrees. So RNA is very unstable. If you keep RNA outside in a test tube, it will degrade in few days. So we keep it at minus 80 degrees in those big freezers, which maintain the temperature as at minus 80 degrees.
Thank you, sir.
Okay.
Hello, sir.
Yes.
So I'm Gauri from JNV Kollam.
Yes.
So telomeres is a hot topic these days. Research are done for anti-aging. What's your view on this, sir? There are cancer can be caused because of its editing. So do you think should we proceed in this side? Should we go for this anti-aging? This concept of immortality is gaining popularity these days.
Yes, so all sorts of research goes on. So there are some scientists who want to develop some ways of anti-aging where you can slow down the aging. And as you said correctly, telomeres are the ones which become shorter and shorter as we age. So that is why people think that by preventing the telomeric shortening, you could slow down the aging process so that we age a little slower and live for more. But that's the research in that area is in very primitive stages. We really are not in a position to really apply that research into actual patients or something. But people are trying now. There is always that, as I said, that ethical dilemma of whether it should be done or not, and that only time can decide whether some things are possible. Whatever is possible, many scientists or people will try to do, but whether it is ethical or not, that is for each of the society to decide. But people are working towards in that particular area.
So I have one more doubt.
Yes.
So we have heard of many dangerous viruses. There are some good viruses also that alter our genes and that help us in a good way now. Why are those not so popular?
No, there are no good viruses. See, all viruses are organisms. Everybody is trying to survive. No, the virus, the bacteria, the fungus, whatever it is, it tries to survive. So for virus, it can only survive inside a cell. Virus cannot survive outside a cell. So it uses people or animals as a host. So it remains in that particular host so that it can divide. So there is nothing called good virus. It is just that during evolution, people feel that these viruses introduce genetic material, and that's how evolution has happened. But whatever virus enters our body and leads to any symptom, that's always a bad virus. So there's nothing good about those viruses.
So is there viruses that are beneficial to us?
No, no, no. There's nothing like good virus or beneficial viruses.
Okay.
Yeah.
Sir, this is Vishnu Priya from JNV Karaikal, sir.
Yes. Yes, go ahead.
Sir, I have a doubt that if a parent has diabetes, is affected by diabetes, sir, after the birth of child, will the child also will have the possibility to be affected by diabetes, sir, or else is there any way to cure that before affected by diabetes?
Yeah. So yeah, that's a good question. So see, we should understand that there are different kinds of human diseases. So there are some diseases which are purely genetic. Okay, that means when there is a genetic mutation, disease happens. When there is no mutation, disease does not happen. And the classical example of that is like sickle cell anemia or thalassemia or cystic fibrosis. These are diseases which are purely genetic. That means mutation is present, disease will happen, and mutation is not there, disease will not happen. But there are other diseases like diabetes, hypertension, coronary artery disease. These are diseases which are called as polygenic or multifactorial diseases. That means they do not happen because of one mutation. So we do not know the genetics of those diseases. We understand very less about genetics of these diseases, and those diseases also need environmental factors for them to occur. That is why if a parent with diabetes gets a child, we can never predict whether this child will get diabetes or not. But people are trying to check for all the genetic factors. When we understand all the genetic factors, then we can predict that this person is more likely to develop diabetes, and we can modify his environment accordingly. So suppose if a person is more likely, then we can tell him to have his lifestyle in a proper way, that have proper diet, don't take a lot of carbohydrates. In that way, you cannot modify the genetics, you can modify the environment. So you can tell lifestyle modification to the people who are at higher risk. So in that way you can do, but at present we cannot tell whether a child will develop diabetes or not in future.
And sir, my grandfather and mother is also affected by diabetes. So my mother is afraid that we will also be affected by diabetes. So she's afraid about
So
it.
yeah, so the risk is there, but it is not necessary. So there are so many families where multiple people are affected and some are not affected. Like maybe in your family also you must be finding that some are not affected, although they have the same genes are shared. Okay, so it is not necessary that everyone will get affected with these particular kind of diseases, but it is always good that you can modify the lifestyle. No, that is always good. You don't need a test to tell you that. So doing good exercise, eating good food, balanced diet, these are all the things you can do, and that can prevent these lifestyle diseases. Okay.
Also, sir, if a baby is in the womb of mother, sir, it is almost 7 to 8 months of pregnancy, sir. If mother is affected by disease like cancer or thyroid, will the baby child will also be affected, no, sir? Is there any way to cure the disease through genetic way?
No, so that's what, if babies, if mother has diseases like cancer or thyroid, that does not affect the same disease in the baby. So baby can get the genetic disease only if mother or father are carriers of genetic mutations. So when the baby is formed, when it is a single stage zygote stage, at that time only it is decided whether baby will get a genetic disease or not. But later on, if mother develops some disease, that may have some effect on the baby's health, but not really leading to any disease in the baby. It may just cause little less blood supply to the baby. Baby may become small or something. Okay, but the genetic diseases come at the zygote level itself, not later on.
Okay, sir. Thank you so much, sir.
Sir, I'm Monica from JNV Chitradurga, sir.
Yes, yes.
Sir, when we go for corona checkup, sometimes they'll take sample from nasal passage and sometimes they'll take sample from both nasal and throat sample. No, sir. Why it's like that, sir?
So the virus is present more in the nasal, the nasopharynx we call it, inside the nose, but
Yes,
it will be present in the throat also. So if they want to be double sure or something, then they must be taking sample from two different locations.
Okay,
So
sir.
from both places you can get the virus and testing can be done.
Okay, sir. Thank you,
Yes.
sir.
Yes.
Hello, sir. Good afternoon, sir.
Yes, yes.
Sir, I am Basuraj from JNV Bijapur, sir.
Yes.
Yes. Sir, my doubt is a brittle bone disease genetic or not, sir? Because some of my classmates are having brittle bone.
I don't know that what your classmates are having, but brittle bone disease is a genetic disease. It is called as osteogenesis imperfecta. The disease name is osteogenesis imperfecta, where even with a small fall, like while playing, if they fall down, they'll get a fracture. Normally, if you fall down or you fight with your friend or something, you don't get a fracture. But these children who have this brittle bone disease, with a simple fall down, they will get a fracture. So multiple fractures they will get. So that is a known genetic disease.
Okay, sir.
Yeah.
Good afternoon, sir.
Yes.
Good afternoon, sir. I am Rajashree from JNV Guntur.
Yes.
So, what is meant by ACE inhibitors?
So ACE inhibitor is something different from the ACE2 receptors that we spoke about for the coronary virus. The ACE inhibitors are basically, ACE means angiotensin converting enzyme. So it is an enzyme which is present in the kidney, and it converts this one molecule called as angiotensin to angiotensin 2. Okay. So these ACE inhibitors are used, these are drugs which are used in treatment of hypertension or kidney failure. So these are different from the ACE2 receptors that we are talking, which is present in the throat of the person for coronavirus. But ACE inhibitors are medicines which are used in treatment of hypertension
Thank you, sir.
Yeah.
Sir, I'm Bhumika from JNV Haveri, sir.
Yes.
Sir, can I know why is PCR used in the process of DNA sequencing, sir?
Yeah, so very good question. So why PCR is needed? So you have seen, you have gone to a drama or a movie, you have seen that the first row people will be able to hear the movie nicely, but the last row people will not be able to hear the movie unless you use a amplifier or a speaker, isn't it? So in the same way, our DNA is so small that it is not even visible with the biggest microscope that we have. It is very, very small. So to make it visible to our eyes, to see the DNA, from one copy, we make millions of copies of that DNA. So once then this DNA is made millions of copies, then we can look at that DNA. So this PCR is used to make large number of copies of this DNA so that we can visualize it, we can see it. Okay. Yes, who else has raised their hands? You can.
Sir,
Yes.
this is Kishore from Jain.
Yes, go on.
Can you explain the endoscopy scan? What is the function of endoscopy scan?
Endoscopy.
Yes, sir.
Where did you hear this? So endoscopy is basically trying to look at the interior organs of the body. So just like you use a telescope to see long distance objects, you see a microscope to see small objects. Similarly, if we have to see inside our body, so inside our body we can either see by doing a CT scan or a MRI. Similarly, we can pass a small tube to see the digestive tract or to see the respiratory tract. You can pass a small tube with a camera, and once this tube goes inside, you can have a look at all the structures inside. So if you want to see the stomach, the intestine from the inside, then you use a endoscope to visualize that. So that is used in diagnosing diseases of the gastrointestinal tract or the respiratory tract.
Sir,
Yes.
why sometimes eye twitching happens?
Yeah, so that's a nice question. So you must have heard about this bad omen or good omen related to this eye twitching, no? So eye twitching is basically a irritation of those muscles, which can happen either due to some small infection there, or if you rub the eyes and some small inflammation has happened. Because of that, the muscles contract. So the muscle contraction and relaxation is felt by us as twitching. You may get this twitching in any other part of the body also. Mostly commonly it happens in the eyes because these eye muscles are very, like, they are in a very thin layer in the eyelids. So any small irritation leads to their twitching. So that's a very normal phenomena. You don't have to feel anything good or bad about that. Okay, but you have a lot of superstitions around this eye twitching and all that.
Is it related to histories?
Related to
Stress.
stress, yeah, sometimes it may happen. So we call that as a psychosomatic. That means our psychological stress, our psychological well-being can affect the somatic, that is the muscles or the physical part. So sometimes stress can lead to this kind of phenomena, but there is nothing to worry about in that.
Thank you, sir.
Yes.
Excuse me, sir.
Yes, go on.
Sir, can you please explain about Huntington's chorea, sir?
Yeah, so Huntington's chorea also is a genetic disease. So this is a type of genetic disease what we call as a late onset neurodegenerative disorder, because here Huntington's disease, the genetic mutation is present right from birth, but the symptoms will come at around 35 to 40 years. So it's called as a neurodegenerative disease. That means slowly the person will keep on losing the different skills that he has. So chorea means abnormal movement. So even if a person doesn't want to move his hand, the hand will start moving. So It's a neurodegenerative disease. Basically, we still do not have any treatment for that, but we can do genetic testing and find out if it is present or absent.
Thank you, sir. 1 minute, sir.
Yes, who is it?
Kishore from Jemipur.
Yes, go on.
Why cannot genetic test predict all diseases?
Yeah, that's what I told you. No, if we understand the genetics of a particular condition, then we can surely predict. But as I told you, there are certain diseases like diabetes, hypertension, coronary artery disease, cancer, where we don't still know all the genetics of those diseases. That is why we cannot predict them. Where we know the genetics of a disease, we can surely predict them. Okay.
Another.
Yes.
Why don't identical twins look same?
What is it?
Why don't identical twins look the same?
Identical twins look the same, no?
Yes, sir.
Identical twins always look the same because their genetic material is exactly the same. So the non-identical twins, they don't look the same because although they are twins, they are like brother and sister. So they will not look the same. But the identical twins are exact. They are developing from the same zygote. The zygote divides into two and then both the cells develop into individuals. So identical twins are always of the same sex and they always look the same.
Okay.
Yeah.
So good afternoon, sir.
Yes.
Similarly, can we identify the chromosomes, sir?
What is it?
Similarly, can we identify the chromosomes, sir?
Where?
Sir is the head of Diagnostic Division, Center for DNA, Fingerprinting and Diagnostic,
We can
Hyderabad.
check for chromosomes. We do testing
And
for
sir
chromosomes.
he's in
So
MD
we take
in
blood sample from the
pediatrics
patients
from Goa
and
Medical
the
College.
WBC is the white blood cells in the
Do your best for whatever
blood
you can
are
do.
then grown in a culture and then
Particular
we
board,
look at the
but
chromosomes
this one
inside those white
mistake in
blood
our
cells.
genome.
So that is done as a
So
genetic testing.
just one
So we have
mistake in
different
3
chromosomal
billion
diseases.
bases.
You
So
must have
such
heard about Down
ATGC, there
syndrome,
are 3
Turner
billion.
syndrome.
But
So
just
these all
one
can
change.
be checked by doing chromosome analysis. So that is done as a test, a genetic test. Yes.
So the question is from JNV Shumoga. Sir,
Yes.
people say that bronchitis is a genetic disorder. Is it true? If it is so, how it will come through genes, sir?
No, bronchitis is not a genetic disorder. Bronchitis means what? Bronchi are the parts of the respiratory system, and itis means it is inflammation. So bronchitis is mostly either post-infective or autoimmune. It is not a genetic disease.
So, but when we go to the doctor, they ask the first question is, do anybody in your family is having the same problem?
Yeah, because see, there are some genetic diseases like cystic fibrosis, or there are other diseases where bronchitis can come as a symptom. Okay, the disease will be something else, but bronchitis is just inflammation of the respiratory passages. So it can come as a symptom. That's why they ask you whether any other person has bronchitis or asthma in the family. So per se, bronchitis itself is not a disease. Okay.
Good afternoon, sir.
Yes.
Daniela from JNV Hassan.
Yes.
Is there any possibility that the two persons other than
Hello.
Can have same DNA?
Yeah. So that's a very good question. So our whole institute is based on this same principle that two persons do not have the same DNA unless they are identical twin, and that is the whole purpose of doing the DNA fingerprinting. You must have heard, no? The DNA fingerprinting is used in criminal cases to catch the culprits. So that is based on this whole principle that two people cannot have the same DNA. That probability is 10 to the power minus millions. So it's like almost impossible that two people will have the same DNA. And that is the whole principle because of which DNA fingerprinting is used by the police and the courts to catch the culprits, because we are sure that the two people are not going to have the same DNA.
Sir, who are test tube babies?
Yes.
Good afternoon, sir. Who are called test tube babies?
Hello. Who? What did you say?
The test tube babies.
So test tube babies is basically when the parents are not able to conceive in the normal way. Then we take the sperm and the ovum, and in the laboratory, the sperm and the ovum are put together to form the zygote. Then this zygote divides for few divisions up to 16 cell, 32 cell. After that, this embryo is then implanted into the mother, and then they can have a normal pregnancy. So such babies are called as test tube babies. So only the initial few divisions are done in test tube. It's not that the full baby is formed in a test tube. This initial few divisions happen in the test tube, and after that the embryo is implanted into the mother, and then like a normal pregnancy, the pregnancy continues.
Good afternoon. My name is
Sir,
class
does
11.
that thing have any side effects on the baby, sir?
What is it?
Good afternoon, sir. My name is
Does test tube babies have any side effects, sir?
class 11, sir.
Yeah,
Is it
so
true that coronavirus is present in everyone's body nowadays, sir? And another question is, how much time does SARS-CoV virus takes to transfer from one person to another person, sir?
yeah, so good questions. So first, I think somebody asked whether there is any side effect of test tube babies. So that is not so true, although some few diseases are known to happen more commonly in these test tube babies, but otherwise world over, so many test tube babies are born, and there is nothing such to suggest that there is any side effect of doing this. So coming to your question, so again, interesting and very important question. So you cannot say that coronavirus is present in everybody's body, because your body is going to destroy or throw away that virus in the few days. So once you develop immunity to that particular virus, that virus is going to die. So it cannot remain in your body for a long time, but the immunity will remain. So your question should be, are all of us immune to the coronavirus? And that is the whole aim of giving these vaccines or getting this immunity through the infections. So hopefully we are all, or we will become all immune to the coronavirus, and that's how then this pandemic is going to end. That is the whole hope that everyone has, that schools will start, colleges will start and all. That can only happen if a large proportion of our population is immune to this virus. So we do not get so many diseases, no, for which we take vaccine, like DPT is there, diphtheria, pertussis, tetanus. These all diseases we don't get nowadays, because vaccine is given during our childhood, and that is how we are able to get immune from these diseases. So same way, we hope that coronavirus also, if everyone is immune, then it will not be spreading from one person to another, and this whole pandemic will stop.
Sir, I am from JNV Institute.
Yes.
Is psoriasis a hereditary disease? What is its cause and remedies?
So psoriasis also is a disease where again is a multifactorial disease. We still don't understand the full genetics of psoriasis. So just like hypertension, diabetes, it is also considered a multifactorial disease. Okay, so it's not a pure genetic disease.
Sir,
Good afternoon.
sir, is there any?
Anurag from Janvi couple.
Yes.
Sir, how transgender are born?
Transgender are a type of, like, what we call is something like ambiguous genitalia. Okay, so our genitals also are part organs
Sir,
of
an
the
echo
body.
is coming.
So hello, yeah, all of you can mute your mic, no, whoever is not talking. Yeah, so see, whenever our body is formed in the mother's womb, so genitals also are one of the organs that develop. So if there is any genetic mutation or any chromosomal abnormality, then these genitalia can be abnormal, and then the society treats them as transgenders. But many of these transgenders are actually not born as transgenders. They are kidnapped or caught while childhood, and then they do castration for them, and that's how they become transgender. So it all depends how a person may be born or may not be born as a transgender.
Okay. Sir?
Okay, sir, thank you.
Yes. Yes.
Sir, is there any change in the genetic code of the baby who is in the surrogacy mother?
No. See, surrogate mother is same like test tube baby, only that you are putting the test tube baby into another mother, not the biological mother. That's all.
Sir, how can we write genetic code, sir?
We
How can
cannot
we know,
write
sir?
genetic code. We can only look at it by sequencing. You sequence the gene, you will know the ATGC sequence of the genes. No, that is what is the genetic code.
Sir, who discovered, sir, this is?
Who discovered what?
Means who said
So
this?
you must have heard about this, no? The Raghavendra Rao. I don't know how many of you
Yes,
have
sir,
heard.
Raghavendra Rao.
Yeah, so Raghavendra
Founded.
Rao was an Indian scientist in USA. He got the Nobel Prize for it also. He
Yes, sir.
discovered the genetic code. That means the 3-letter code which amino acid is coded by a particular codon.
Sir, you only discovered, no, sir, why the cancer is caused.
What is it?
Sir, that Hargobind Corona only discovered, no, sir, why the cancer is caused.
Not about cancer. He discovered the genetic code, like what is the like ATG codes for methionine, like that. What is the code for each amino acid? That's what is credited to him. But he must have worked on many other things. So a scientist doesn't work on only one thing, no? So they work on multiple things. So all those things are also there. Yeah.
Sir, how long does COVID-19 last on surfaces, sir?
What is it?
How long does COVID-19 last on surfaces?
Yeah, so that again is a
But it is
question
going
which
to death,
many people are not very sure about, but people feel that the virus may survive up to it becomes dry. See, basically the virus cannot survive outside the cell. So if the virus falls on a inanimate object and it dries off, then the virus will not survive. If the virus is able to find a cell, it will enter the cell and then it can survive. Okay.
Sir, what is the use of remdesivir?
So remdesivir, you must have heard about this drug, no? So that drug is mainly used, it is an antiviral drug actually. So it is mainly used to, it can only help to prevent severe infection from happening. So if you give remdesivir in the very early days of infection, then it can prevent it from becoming severe, but it cannot totally cure the virus. So that is why it is mostly now used only in people with comorbidities and all. No, everybody it is not given.
Sir, what it will do, sir, if it goes inside the body?
What is it?
Sir, remdesivir goes into the body, what it will do, sir?
It will go and kill the virus. No, it is antiviral. It goes and kills the virus, but it has a lot of side effects also. That's why routinely it is not used for everybody.
Yes, sir. During COVID, it has raised the cost also, sir.
Yes, because it is less available, no? So.
Excuse me.
Yeah. Yes. Please one at a time, please. Yeah.
Excuse me, beta. Please let me interrupt. Actually, the time is almost over, right? Next session we have at 5 o'clock. So still, if you have some queries, I'll request Dr. Ashwin sir to please drop your mail ID if it's okay, sir.
Yes.
Ma'am, have you sent the attendance on chat box?
Yeah, attendance will be there.
Ma'am, but we don't have the chat.
So I think chat is not available, I think, for. But you can anyway. You have my email address, so you can send my email address to all of them. Anyone can send an email to me. I'll be happy to answer.
Sure.
Yeah.
Sir, now we have our Suresh sir, our PGT Bio sir in your chamber, maybe, sir. So he came there to facilitate you, sir.
Madam, I am here only.
Yes, sir. Yes.
Manju madam, can you hear me?
Yes, sir, Suresh sir.
Yeah, I am here only. Shall we begin that?
Yeah.
Shall I?
Please come in the picture, Suresh sir. If it's possible.
Yeah, you can come this.
Yeah.
Sir, I have a doubt, sir.
Wait a minute, beta. Wait a minute.
Oh.
Yeah, thank you so much, Ashwin sir. And it was really a pleasure that Navodaya alumni. Thank you so much, sir. So every session is interactive. If you have any more doubts, students, you can send the mail to sir. We will definitely share the mail ID of Ashwin sirs. Okay.
Okay, ma'am.
Okay. Ashwin sir, can we wind it up now?
Okay. Yeah. Thank you. Thank you very much.
Okay. Thank you so much.
Yeah.
Thank you, madam.
Ma'am, attendance.
1 minute, beta.
So I'll leave the meeting.